Hello, good day. We are your histopathologists and histotechnicians for today, and we will be demonstrating the proper steps in performing some of the methods in tissue processing, of which are infiltration, embedding, fishing out, H&E staining, and mounting. Infiltration. Infiltration, also known as impregnation or interpenetration, is the saturation of tissue cavities and cells by a supporting substance, which is generally, but not always, the medium in which they are finally embedded. In this procedure, we need paraffin wax, paraffin dispenser, paraffin oven, glass dish, and cleared tissue samples. For this demonstration, we will be infiltrating the tissue specimen by using wax. We will be immersing the tissue specimen into 3 separate paraffin wax baths in the proper order and time interval. Immerse the tissue specimen in the first paraffin wax bath for 30 minutes. After that, we will place the tissue specimen in the second paraffin bath for another 30 minutes. Finally, we will be placing the tissue specimen in the third paraffin bath for 45 minutes. Lastly, remove the tissue samples and place on paper towels. Embedding. Embedding, also known as blocking or casting, is the process by which tissues are surrounded by the infiltrating medium, which, when solidified, provides sufficient external support during sectioning. It is usually done after infiltration. Characteristics of an ideal embedding media are the same as those for infiltration. In this procedure, we need the following resources: infiltrated tissue sample, paraffin wax. Make sure that the paraffin wax is melted and the dispenser is full before performing the procedure. Paper boat, forceps, and cooling tray. Now, we will proceed to the embedding proper. After the last paraffin wax bath during infiltration, prepare the embedding molds. Get an infiltrated tissue sample from the tissue cassette. Remember to attend to 1 sample at a time and strictly 1 mold per tissue only. Dispense a small amount of paraffin to act as a base and immediately transfer the tissue into the central portion of the mold. Observe proper tissue orientation. The greatest factor to consider during embedding is the orientation of the tissue specimen. Careful specimen orientation in the mold determines the plane of section. If not all layers are exposed, the hidden layer cannot be examined. Then, the diagnosis may be wrong. Next, fill up the rest of the mold with paraffin wax. Repeat the process until all tissues are in a mold. Place the molds on a cooling tray to solidify. Once done, detach the block from the mold and then label. Fishing out. Tissue flotation bath is required after the step of cutting paraffin sections and before these are placed on slides. It lets tissues relax and smoothen before being mounted on a slide, as well as makes paraffin stick to the slide. This ensures removal of wrinkles and folds before sections are placed on the slide. In fishing out procedure, we will need the following resources: glass slide, flotation water bath, onion, and oven. In fishing out the onion tissue ribbons, the first thing to do is place the slide vertically near the target tissue section. It is important to maintain the temperature of the water between 5 to 9 degrees Celsius below the melting point of the wax, commonly around 40 to 60 degrees Celsius. Float out the section for approximately 30 seconds to flatten and avoid overexpansion of the slide. Then, fish out with a gentle pulling action to avoid wrinkling and crumpling the section. Then, after fishing out the specimen, place the tissue slides in the oven set at 60 degrees Celsius or 5 to 10 degrees Celsius above the melting point of the wax for about 30 minutes. H&E staining. Hematoxylin and eosin staining is the most common staining method in routine tissue processing. It provides an effective means of staining different cellular components to a wide variety of tissue types. It uses hematoxylin as a primary stain and eosin as the counter stain. These are the materials needed for routine H&E staining: tissue sections, staining jars and dishes, beakers or glasses, staining racks and trays or plates, xylene, alcohol formol, 95% ethanol, hematoxylin, eosin Y, acid alcohol, ammonia water, dishwashing soap, cotton swab, paper towels, and flotation water bath. Carefully fish out the section specimen, following the correct method as formerly demonstrated. Then, dry in an oven with a temperature 5 to 10 degrees Celsius above the melting point of wax for 30 minutes. Or, air dry for several hours or overnight. After drying, the actual staining procedure will be started. First is deparaffинization. Immerse the slide in 2 changes of xylene for 5 minutes each. Xylene will remove the paraffin from the section, since paraffin is insoluble with the aqueous stains that are commonly used. After deparaffinization is the hydration step, which starts with 10 dips in alcohol formol or absolute alcohol. Alcohol is miscible with both xylene and water and is therefore effective in removing xylene from the section. Next is to immerse the section in 3 changes of 95% alcohol with 10 dips each change. We can also use a series of aqueous alcohol in descending concentrations for delicate specimens. Rehydrating the section in a descending alcoholic concentration will protect the tissue from damage and hydrate the section effectively. After hydration, wash the slide in soapy water and remove excess albumin adhesives using a cotton swab. The section must not be touched by the cotton swab. Cleaning the slide before the actual application of staining solutions is a must in order to avoid coloration of the slide, contamination of the specimen, and avoid artifact formation by the reaction of the adhesive on the free spaces of the slide. Rinse the slide in clean water and drain excess water using paper towels. After washing, immerse the slide in hematoxylin for 5 to 10 minutes. Hematoxylin is a basic dye which stains acidic components of the cell, such as nucleus, imparting a dark blue or purple color. Hematoxylin is usually mixed with a mordant, commonly aluminum, and oxidized to become a more effective hematine by adding oxidizing agents. Drain excess stains and rinse with water. After 5 to 10 minutes, use acid alcohol as a decolorizing agent by quickly dipping the slide twice to minimize damage by the reagent. Decolorizers will remove excess hematoxylin stain, enabling differentiation by the counterstain eosin Y. Rinse immediately with water. Next, immerse the slide in ammonia water for an average of 5 minutes for bluing. Ammonia will provide an alkaline environment for the hematoxylin-soluble red color to become insoluble blue, making the stain permanent. Rinse with water and drain excess water with paper towels. For the counter stain, immerse the slide in eosin Y for 1.5 to 3 minutes. Eosin Y is an acidic dye that stains the basic components of tissues such as the cytoplasm and connective fibers. These structures will appear shades of orange, pink, or red, contrasting the purple-blue complex by the hematoxylin. Rinse and drain excess water. Next, dehydrate the section in an increasing concentration of alcohol or 3 changes of 95% alcohol or ethanol with 10 dips each. This will remove excess water from the section, making it compatible with the clearing agent xylene and subsequently with the mounting media. After dehydration, clearing with 3 changes of xylene with 10 dips each change should be done to replace alcohol and make the section translucent. Drain excess xylene and proceed to mounting. Mounting. Mounting is the last procedure in the histology or pathology section. It ends with a permanent histological preparation. This procedure is important since it protects the stained tissue section from getting scratched and from bleaching or deterioration. We must utilize a mounting medium or a mountant during this procedure. It enhances the image quality of the sample, thus we can appreciate the microscopic details of the tissue. In this process, we will be using glass slide with the stained tissue section, cover slip, and of course, the mounting medium. Once the materials are ready, we will start mounting. First, grab a cover slip and add a drop of a mounting medium. For the purpose of this return demo, we will just use an oil. After this, we will grab our glass slide containing the mounting medium and rub its one end on the cover slip. Now, we will gently push the cover slip into the glass slide and make sure that there are no air bubbles that will form. Next, tap the glass slide to remove the excess mountant and wipe the back portion of the glass slide to make it clean. Then, repeat the procedure for the next tissue sample section.